Usually, when we talk about getting a medical diagnosis, there's this expectation of like mechanical precision.
Right, yeah.
You know, you break your arm, the x-ray shows a jagged white line, and the doctor just points to the screen. It's broken or it's not broken. It's a clean binary system. But the second you step into the world of tumor biology and you look at this microscopic battlefield inside the human body, that whole x-ray machine metaphor just, it shatters.
It really does. It falls apart completely.
The landscape we're looking at is it's completely wild, it's adaptive, and honestly, highly deceptive. So, welcome to our deep dive. Today we're exploring a groundbreaking 2025 paper from Nature Cancer that fundamentally rewrites the rules of engagement for how tumors grow.
Yeah, we're going straight to the front lines of this microscopic war zone today.
Exactly. And our mission for you, the listener, is to unpack this brand new discovery, detailing exactly how cancer cells physically hijack the healthy tissue surrounding them.
And, you know, the traditional view of cancer has always been somewhat passive. Like, it's just this rogue cell that endlessly divides and crowds out healthy tissue.
Just taking up space, basically.
Right. But this study forces us to look at the tumor microenvironment, or the TME, as a highly active, totally corrupted ecosystem. A tumor isn't an isolated island of bad cells. It's embedded in this scaffolding of healthy cells. And the most critical players in that scaffolding are the fibroblasts.
Okay, let's unpack this role of the fibroblasts for a second so everyone is on the same page. You can think of them as the biological architects or like the construction workers of our connective tissue.
That's a great way to put it.
Right. Like they're just innocent bystanders whose normal day job is maintaining the structure of your skin, your organs, your tissue matrix.
Just doing their job.
But in the context of cancer, these builders get brainwashed. They get converted into what researchers call CAFs, cancer-associated fibroblasts.
Yes, CCAFs.
And up until now, the prevailing theory was that this brainwashing happened essentially wirelessly. We thought the tumor was just secreting these toxic chemical signals into the surrounding fluid, you know?
Yeah.
And the fibroblasts were just passively absorbing those chemicals and going rogue.
Which, to be fair, made perfect sense based on the tools we had to observe them at the time.
Right.
But this paper reveals a mechanism that is far, far more invasive. Cancer cells are not just sending, like, chemical text messages.
No, it's so much worse.
It is. They are actively transferring their own powerhouses, their mitochondria, directly into the normal, healthy fibroblasts to force that cellular corruption.
Okay, let's unpack this, because it gives me serious Borg assimilation vibes from Star Trek.
Yes.
I mean, they are physically implanting their own microscopic hardware into a healthy cell, literally stripping it of its original programming to assimilate it into the collective.
That analogy actually captures the severity of what's happening perfectly. They are implanting metabolic hardware. And the implications are massive here because historically, the scientific literature showed the exact reverse.
Wait, really? The reverse?
Yeah. We knew that healthy stromal cells or immune cells sometimes sacrifice themselves. Like they would donate their mitochondria to cancer cells to help the tumor survive a harsh, nutrient-deprived environment.
Oh, wow. So the healthy cells were acting like a battery pack for the tumor.
Exactly. It was framed as healthy tissue being tricked into feeding the tumor. But this paper flips the script entirely.
The cancer cells are deliberately giving away their own precious energy factories to weaponize the healthy fibroblasts.
Yes, exactly.
Which raises an immediate glaring question for me. If they aren't just floating chemicals across a gap, how are they actually pulling off this physical hardware transfer?
It's a logistical nightmare, right?
Totally.
Right.
I mean, a mitochondrion isn't some small molecule. It's a massive, complex organelle. How'd you get that from one cell to another without just destroying both cells in the process?
Well, the researchers identify the physical infrastructure the cancer uses. They are called tunneling nanotubes, or TNTs.
TNTs?
Catchy. Right. These are incredibly thin, elongated, membranous bridges that actually connect two distinct cells.
How thin are we talking?
To give you a sense of scale, these tubes are about 10 to 100 micrometers long, stretching right across the extracellular space.
Okay.
And they are constructed primarily out of an internal skeleton of actin filaments.
So it's like a physical rope bridge made of actin.
Exactly. And the researchers literally observed these TNTs forming direct physical pipelines between cancer cells. Specifically, they looked at A431 vulvar carcinoma cells, MDA-MB231 breast cancer cells, and PANC1 pancreatic cancer cells.
Okay, so multiple types of cancer.
Right, multiple types. And they watched them build these bridges straight into human primary skin fibroblasts, or HPFs.
Okay, I had to play the skeptic here for a minute because of how these microscopic imaging studies are usually done.
Go for it.
I read the methodology, and initially the researchers used fluorescent dyes, specifically one called Mitotracker Green, right?
Yeah, that's standard practice.
They used it to make the cancer mitochondria glow so they could track their movement. But if I'm looking at a glowing green dot inside a healthy fibroblast, my first thought is dye leakage.
Ah, the classic confounding variable.
Exactly. Couldn't the dye simply be seeping out of the cancer cell, drifting across the fluid, and just passively staining the fibroblast's native mitochondria? I mean, dye leakage is a notorious problem in these types of studies.
And the researchers anticipated that exact skepticism. Their methodology to rule out passive leakage is what makes this Nature Cancer paper so robust.
So how did they prove it wasn't a leak?
They abandoned the chemical dyes entirely and moved to genetic hard coding.
Oh, wow.
Yeah. They engineered the human fibroblasts to permanently express a red fluorescent protein targeted right to their mitochondria. They used a marker called Si9RFP for that. And concurrently, they engineered the cancer cells to permanently express a green fluorescent protein in their mitochondria using a marker called So9GFP.
So the host tissue is permanently red, and the invading cancer hardware is permanently green.
Yes. So when they observed the co-culture under a confocal microscope after 24 hours, they didn't just see green dots floating around.
What did they see?
They saw that the fibroblasts contained mitochondria that appeared orange.
Oh, because the red and green physically combined?
Exactly. The green hardware from the cancer cell had physically crossed the cellular divide and literally fused with the red mitochondria of the fibroblast.
That is insane. You definitely cannot fake a fluorescent color fusion just by having some dye wash over the cell.
No, you can't. But, they actually pushed the proof even further.
Because fluorescence alone still leaves, like, a tiny margin for visual error.
Right. So they tracked the DNA itself.
Oh, wow.
They set up a cross-species experiment, which is where the data becomes just absolutely undeniable.
Cross-species?
Yeah.
Like human and animal?
Yes. They mixed human A431 cancer cells with mouse fibroblasts.
Okay, so human cancer, mouse healthy tissue.
Exactly. And after a 24-hour incubation period, they had to isolate the mouse cells to see what was inside them.
How do you separate them back out?
They looked for a specific protein called fibronectin-1, which was exclusive to the murine or mouse cells in this setup.
Got it.
And once they had those isolated mouse fibroblasts, they sequenced the genetic material inside them.
And let me guess, they found human DNA inside the mouse cells.
Not just any DNA. Specifically, they found unique human mitochondrial DNA.
Unbelievable.
They tracked species-specific single nucleotide polymorphisms, or SNPs.
Oh, right, SNPs. I've heard of those.
Yeah, they're essentially just single-letter typos in the genetic code. And these specific SNPs were entirely unique to the human 16S ribosomal RNA gene region.
Okay, so finding those human SNPs inside a mouse cell absolutely confirms that the physical organelle carrying that DNA made the journey.
It's the ultimate biological fingerprint.
So the hardware is undeniably moving across. But to prove that those actin bridges, the TNTs, were the actual delivery mechanism, the researchers started playing with the cell's construction equipment, didn't they?
They did. They introduced a drug called dihydrocytocalcin B.
Okay, wait. Let's create a quick mnemonic for the listeners so we don't get lost in the jargon. Let's go with A for actin, A for action.
I like that. A for actin, A for action.
Because dihydrocytocalcin B blocks the action of actin, right?
Yes. It's an actin polymerization inhibitor. See, actin normally forms by stacking individual proteins, kind of like Lego bricks, to build a long scaffold. This drug basically caps the filament. It completely stops the cell from stacking any more Legos, meaning it can't build those actin-based nanotubes.
And when they blocked the actin construction, the transfer of mitochondria completely halted.
Exactly.
The bridges couldn't be built, so the cargo couldn't move. But the part of the study that really caught my attention was their use of a second drug. Yes, carboxylone. Because this drug blocks gap junctions, which are those simple, passive little pores that naturally exist between neighboring cells.
Right, the normal communication channels.
Exactly. Now, you would assume that if you block any kind of connection between cells, the transfer of mitochondria would drop, right?
That would be the logical assumption.
But the addition of carboxylone actually caused the mitochondrial transfer to increase.
Yeah, that seemingly contradictory result is just a beautiful piece of scientific deduction.
How so?
Well, think about it. If you block the passive pores and the transfer goes up, it proves that the mitochondria were never using those pores in the first place.
Oh, because they're too big anyway.
Right. And by shutting down the gap junctions, the cancer cells were likely forced to panic compensate.
Like, our normal comms are down, build more bridges.
Exactly. They upregulated the construction of the active, complex actin nanotubes. It proves the transfer relies exclusively on the cell actively building this specific infrastructure.
Wow. Okay, so time for a quick recap for you, the listener, before we move to the next step. So bullet point 1. Cancer cells build these physical actin TNT bridges across the microscopic void. Bullet point 2. They pump their own mitochondria across these bridges. And bullet point 3. This was irrefutably proven by tracking human DNA inside mouse cells.
That's the perfect summary.
It really makes you pause and consider the implications for our own biology, though. I mean, think about it. If a rogue cancer cell can build a physical bridge on demand to share complex hardware, what kind of information or organelles do you think your own healthy cells are sharing right now, deep inside your tissues, to keep you functioning?
The interconnectedness is just staggering when you really visualize it.
It really is. But returning to the tumor, we have to look at the fallout of that connection. We know the engine has been shipped to a new garage. The next logical question is, how does this transplanted hardware actually take over the driver's seat and brainwash the fibroblast?
Yeah, step 2. To figure out exactly what the mitochondria were doing once they arrived, the researchers had to completely remove the cancer cells from the equation.
Because of the confounding variables again.
Right. If the cancer cell is still sitting there in the petri dish, you don't know if the fibroblast is reacting to the transplanted mitochondria or just reacting to some random chemical the cancer cell is still secreting.
Right, you have to isolate the variable.
Exactly. And to do this, they utilized a technique called mitoception.
Mitoception. Honestly, it sounds like a sci-fi thriller where we're planting ideas into people's dreams, but like on a cellular level?
It kind of is.
How do you manually force a mitochondrion into a cell?
It's an artificial transplantation protocol.
Okay.
The researchers took the cancer cells and mechanically broke them open. Then they extracted and purified just the mitochondria.
Just the engine.
Just the engines. Then they took these isolated cancer mitochondria, placed them onto cultures of healthy human primary fibroblasts, and used very specific centrifugation and buffer conditions to basically force the fibroblast to internalize them.
So they artificially recreated the end result of the nanotube transfer, but in a completely sterile, controlled environment.
Exactly. It was just the innocent fibroblast and the newly introduced cancer mitochondria. Nothing else.
And the resulting metabolic shift was massive, according to the paper. It highlights that these cancer mitochondria are not just normal, everyday energy factories. They are metabolically distinct.
And highly aggressive.
Highly aggressive. How do they measure that aggression?
They use something called a seahorse analyzer.
A seahorse analyzer.
It's a specialized piece of equipment that measures real-time cellular metabolism. It does this by tracking how fast cells are literally consuming oxygen.
Oh, wow.
Yeah. And they found that the oxygen consumption rate, the OCR, and the basal respiration of the recipient fibroblast just absolutely skyrocketed.
Meaning they were burning through fuel.
Right. This forces the fibroblast into a state of high oxidative phosphorylation, or OXPHOS. The cancer mitochondria are fundamentally rewiring the energy grid of the host cell.
Okay, here's where it gets really interesting. To visualize this, imagine the healthy fibroblast is a sensible, reliable commuter sedan.
Okay, I like this.
It wakes up, it maintains the connective tissue, it does its job quietly and efficiently. Then mitoception happens.
The infiltration.
Right. It's the equivalent of someone sneaking into the garage in the dead of night and swapping out the sedan's sensible engine for a massive, fuel-guzzling, hyper-aggressive race car engine.
That is exactly what's happening energetically.
Suddenly, the sedan is forced into absolute overdrive. The massive spike in energy production causes the fibroblast to multiply at an unnatural rate. And more importantly, it forces the cell to begin secreting a cocktail of tumor-promoting proteins.
The researchers call that a protumorigenic secretome.
A protumorigenic secretome. The sensible sedan has been turned into a getaway vehicle for the tumor. It is officially a cancer-associated fibroblast.
But what's fascinating here is the necessity of that race car engine being fully operational. That was a critical point the researchers really wanted to verify.
Oh, right. Because it's entirely possible that the fibroblast wasn't being driven by the energy of the mitochondria.
Right.
It could have simply been triggering a panic response because it detected foreign biological debris.
Exactly. They needed to prove that the engine's function was the actual cause of the brainwashing, not just its physical presence.
Right, because if you put a broken engine into the sedan, and the sedan still speeds up, well, then the engine isn't the thing causing the acceleration.
Right, it would just be a panic response to the mechanic being in the garage.
So did they try sabotaging the mitochondria before transplanting them?
They did, using two distinct methods. First, they used a chemical called CCCP.
What does that do?
It depolarizes the mitochondrial membrane.
Okay, break that down for us.
Sure. Mitochondria generate energy by maintaining a very delicate gradient of protons across their inner membrane. It's like water behind a dam. CCCP destroys that gradient. It effectively ruins the mitochondrion's ability to generate ATP, which is cellular energy. It breaks the dam.
Got it. And this second method?
For the second method, they used cancer cells that were entirely depleted of their mitochondrial DNA. These are known as LMT cells.
So they essentially wiped the hard drive.
Exactly. So they took these sabotaged broken engines and forced them into the fibroblasts using that mitoception technique again.
And what was the reaction this time?
There was no reaction.
Wait, really? Nothing?
Nothing. When the depolarized mitochondria, or the ones lacking genetic instructions, were introduced, the fibroblasts remained completely normal.
Wow.
Their proliferation rates didn't spike, they didn't switch to high oxidative phosphorylation, and they didn't release that toxic tumor-promoting secretome.
That perfectly isolates the mechanism. The brainwashing strictly requires a functional power source.
And then the researchers took this sabotaged engine model and tested it in live mice. This is where the clinical stakes become incredibly clear.
Okay, let's hear the mouse data.
They injected mice with tumor cells alongside fibroblasts. Some fibroblasts had received functional cancer mitochondria, and some had received the broken ones.
And let me guess, the tumors with the functional cancer mitochondria grew aggressively.
Extremely aggressively. But the tumors grown alongside the fibroblasts with the broken mitochondria, they stayed significantly smaller.
Wow.
The tumor's ability to grow in a living organism is directly tied to its ability to outsource its metabolic demands to the surrounding tissue using fully operational hardware.
It's literally treating the surrounding tissue like a server farm, hijacking its processing power to run its own malignant software.
That's a great analogy.
So let's do another quick recap. Functional cancer mitochondria enter the fibroblast. Metabolism spikes. Fibroblast turns into a tumor-helping CF.
Right.
But think about the inverse of this for a second, listener. If cancer mitochondria are so high performance that they can force a resting cell into energetic overdrive, what do you think would happen if we could capture that capability?
Oh, you're talking about regenerative medicine.
Exactly. Imagine harnessing those hyperactive engines and delivering them into a cell that is actively dying from a lack of energy, like a failing heart muscle cell after a heart attack. The exact same mechanism destroying tissue here could theoretically be used to resurrect it elsewhere.
It's a profound thought.
It really is. But returning to the tumor's logistics for now, we have a clear picture of the bridge, the actin TNTs, and we know the cargo, the hyperactive mitochondria. But organelles don't just magically float to the edge of a cell and jump across a bridge. They had to be actively driven there.
Right. We need to look at the logistics commander inside the cancer cell making this happen.
Step 3.
Yeah.
Who is driving the delivery truck?
The paper pinpoints a specific genetic commander for this entire operation. It's a protein called MIRO2.
MIR2?
Yeah. MIRO2 is a mitochondrial trafficking protein.
How does it work?
Well, inside a cell, mitochondria are constantly being moved around on this internal highway system made of microtubules.
Okay, so there's a literal road network inside the cell.
Exactly. And the mitochondria are pulled along these highways by cellular motor proteins. MIRO2 acts as the crucial adapter molecule that links the mitochondria to the motor protein.
Oh, so it's the hitch that connects the trailer to the truck.
Yes. It is the delivery driver.
So what happens if you take the driver out of the equation?
The researchers did exactly that. They used gene silencing techniques to suppress the expression of the MIRO2 gene in the cancer cells, and the entire assimilation process just collapsed.
Without MIRO2 acting as the logistical tether, the mitochondria couldn't navigate the cell.
Nope. They just clumped up in this useless pile around the nucleus at the center of the cancer cell.
Oh, they got stuck.
Right. Because they couldn't reach the outer plasma membrane, they obviously couldn't enter the tunneling nanotubes. The transfer dropped to near zero, the neighboring fibroblasts remained completely uncorrupted, and in the mouse models, the tumor growth essentially hit a brick wall.
Wow. So the tumor's progression is absolutely dependent on MIR02 orchestrating the movement of these organelles to the periphery of the cell.
100%.
Okay. But as I was reviewing the extended data in the paper, specifically Figure 8, I hit what looked like a massive contradiction.
I know exactly what you're going to bring up, the survival plots.
Yes, the Kaplan-Meier survival plots for human patients. They looked at several different cancer types. Now, normally, if you find a master villain protein, like MIR02, that is driving the cancer's spread, you would expect the data to show that human patients with the highest levels of that protein have the lowest survival rates.
Right, that's the standard assumption.
But if you look at the global bulk levels of MIR02 in these human tumors, high MIR02 expression doesn't broadly correlate with worse overall survival.
It doesn't.
How can MIR02 be the linchpin of tumor invasion if patients with lots of it aren't actually dying faster?
That paradox is exactly what forces us to evolve how we measure tumor biology. For decades, the standard method for analyzing a tumor's genetic profile was essentially throwing the entire tissue sample into a blender.
A blender?
Metaphorically speaking. You grind up the whole biopsy, extract the RNA, and get a single average measurement of what genes are turned on.
Oh, I see.
It's like trying to understand the traffic patterns of New York City by looking at the total emissions of the entire state of New York. You lose all the detail.
Right.
The researchers resolved the MIR02 contradiction by abandoning the blender approach and using something called advanced spatial transcriptomics.
Spatial transcriptomics. Meaning, instead of a biological smoothie, you are creating a high-resolution satellite map.
Exactly.
You're taking a physical slice of the intact tumor and tagging exactly where specific genes are active, cell by individual cell, while preserving their geographic location in the tissue.
And that is the crucial difference. Because the spatial map revealed that MIR02 is not uniformly turned on across the entire tumor.
Where is it?
Well, in the dead center of the tumor mass, MIR02 levels are actually quite low. But at the absolute outer boundaries, what we call the invasive leading edge MIR02 is massively overexpressed.
Okay.
The researchers identified a specific subpopulation of cells at this edge called tumor-specific keratinocytes, or TSKs.
They are the vanguard troops.
Yes.
Think about it. The cells trapped in the middle of the tumor, they don't need to build bridges and export mitochondria because they are already completely surrounded by other cancer cells. There's no healthy tissue left to brainwash in there.
Exactly. It's only the vanguard cells on the absolute front lines physically touching the healthy connective tissue that need to crank up their MIR02 production.
They needed to deploy the hardware and carve a path forward.
Right. The deployment of MIR02 is highly tactical and extremely localized. When you grind up the whole tumor in that blender, the massive volume of quiet cells in the center completely drowns out the screaming signal of the vanguard cells at the edge.
But spatial transcriptomics caught them red-handed at the border.
It did.
Okay, recap number 3 for you, listener. MIR02 drives mitochondria to the edge. It's concentrated exclusively at the tumor's invasive front, and blocking MIR02 stops the brainwashing and halts tumor growth.
Beautifully summarized.
It changes everything about how we view the physical structure of a malignancy. Think about spatial awareness in medicine, listener. How might targeting just the edge of a tumor change how we design cancer drugs? We don't necessarily need to poison the entire mass if we can just build a molecular fence that neutralizes the vanguard's ability to use MIR02.
It creates an incredibly precise target for intervention, especially for stroma-heavy malignancies.
Like which ones?
Pancreatic cancer is a great example. It is notoriously lethal precisely because it builds an incredibly dense fibrotic wall of these corrupted CAFs around itself.
Like physical armor.
Exactly like physical armor. It prevents chemotherapy drugs from reaching the tumor cells inside, and it hides the tumor from the patient's immune system.
But if we can develop an inhibitor that specifically targets MIR02,
we cut off the cancer's ability to recruit those builders.
Yep.
We strip the tumor of its armor, exposing it to conventional therapies.
that is just incredible. So what does this all mean, big picture? We've gone from looking at a static lump of rogue cells to observing a highly coordinated, invasive architect. The cancer builds physical actin bridges. It ships its own hyperactive metabolic engines across the void. And it relies on precision logistics proteins to push that cargo to the front lines, turning innocent scaffolding cells into complicit armor builders. It is terrifyingly elegant.
It really is. But, as we synthesize all of this, there is a much deeper, almost philosophical biological truth buried in the literature surrounding this paper.
Oh, what's that?
Intercellular mitochondrial transfer via nanotubes is not a mechanism that cancer invented.
Wait, really? Cancer didn't evolve this trick?
No, it didn't. The researchers point out that mitochondrial transfer is a deeply ancient evolutionary mechanism, and it's normally utilized for tissue revitalization and healing.
Healing.
Yeah. In a healthy human body, when an organ experiences severe injury or oxygen deprivation, like, say, during an ischemic stroke,
Right.
healthy stem cells will naturally construct these exact same actin nanotubes. They will deliberately transfer their healthy mitochondria into the dying cells to rescue them from apoptosis or cellular death.
So, cancer essentially stole the blueprint for our body's emergency medical response.
They did.
It took a lifeline designed to save dying tissue and weaponized it into an infection vector to spread corruption.
It is the ultimate biological hijacking. The tumor didn't invent the network. It simply learned how to exploit the administrative privileges of a system that was already built into our connective tissue.
Wow, that reframes everything.
Well, yeah.
And it leaves us with an incredibly profound question to consider as we wrap up. If cancer is just exploiting an ancient, hidden cellular internet, using actin bridges to essentially pass out USB drives loaded with malicious code,
Right.
it makes you wonder about the broader landscape of human disease. What other chronic conditions might be secretly using this exact same dark web?
That's the million-dollar question.
Right. I mean, listener, mull this over. Could the misfolded proteins in Alzheimer's disease or the persistent dysfunction in autoimmune disorders be utilizing these hidden physical pipelines to spread entirely off the radar of our immune system?
It suggests that the future of medicine won't just be about targeting bad cells, but about policing the hidden infrastructure that connects them.
If we can map this underground network, we might not just defeat tumor growth. We might uncover the hidden highways driving the most complex diseases of aging.
Absolutely.
We started today looking at a murky diagnostic landscape. But thanks to the rigorous work in this Nature Cancer paper, those waters are clearing to reveal a cellular world that is more connected, dynamic, and breathtakingly complex than we ever imagined.
It's an exciting time for biology.
It truly is. We want to thank you for joining us on this deep dive into the microscopic front lines. Until next time, stay insanely curious.